All right. Hello, everyone, and welcome to today's webinar, Lipidomics, Shotgun Lipidomics, and Functional Lipidomics for Alzheimer's Research. I'm Tyrone Lawton from the Publication Department at the American Society for Biochemistry and Molecular Biology, ASBMB, and I'm thrilled to be your moderator today. This webinar is part of a breakthrough webinar series sponsored by the ASBMB journals. Specifically, today's session will be brought to you by Journal of Lipid Research, JLR, one of our esteemed ASBMB journals, which has been at the forefront of lipidomics and metabolism research for over 60 years. Before we dive in, a quick reminder, you're welcome to submit questions at any time using the Q&A feature at the bottom of your screen. We'll address them during the dedicated Q&A session after the presentation. This webinar is being recorded, and the recording will be available on the ASBMB website. It is now my pleasure to introduce today's speaker, Dr. Xianlin Han. Dr. Han is a chair professor of medicine at the Boshoff Institute University of Texas Health San Antonio. He graduated from Zhejiang University and completed his graduate and postdoctoral training in the lab of Richard Gross at Washington University School of Medicine. Dr. Han has published over 400 papers with more than 40,000 citations. He is one of the pioneers in lipidomics and associate editor of the Journal of Lipid Research, and has received numerous awards while serving in NIH study sections. His broad interest in functional lipidomics continue to shape the field. Today, Dr. Han will be sharing insights into his work on shotgun lipidomics and how this approach enhances our ability to analyze complex lipid profiles. Without further ado, please join me in welcoming Dr. Han.
Thank you, Tara. Thank you for your kind introduction, and also thank the ASBMB to organize this webinar series. And thank you all for your interest into my presentation. So today I'm going to take this opportunity to discuss lipidomics, shotgun lipidomics, and functional lipidomics briefly, and some work we are done in these areas, as well as share some of our findings on Alzheimer's research by using the functional lipidomics approach. So to this audience, I believe majority of us know is lipids are very important. It's a very important component in the cells and play many roles. And shown here is the three essential roles for lipids in the cells, including the, let me turn on the, please. So, including the membrane structure, signaling, and the bioenergetics. Of course, no definitive kind of way can define which class of lipids plays which role. So more or less kind of cross over. And in addition to the cellular function, of course, there existing many extracellular functions, such as signaling and interactions with other cells, as well as proteins or something. And in addition to such an important role, and the lipids we know also very complicated. And show here as an example, about 20 years ago, the Lipid Map Consortium classifies the lipids into 8 categories of the lipids shown here. I just would like to give you an example about the glycerol phospholipids, how complicated it is, and we understand it for that. So here is a simple way to illustrate this category of lipids. Of course, it looks like it's very simple, and the center is glycerol and contain phospholipids, but it contains 3 variables, X, R1, and R2. So to make this category of lipids very, very complicated. And the X is the small molecules, polar molecule defines the class of glycerol phospholipids. And R2 usually is a free fatty acid and contain different number of carbons, double bonds, as well as double bond locations. And the SN1 position, R1, is more complicated. In addition to this third type of the aliphatic chain, and also has 3 different connections. So vinyl ether connection, and the esters. So you can easily calculate this category of lipids containing at least like 30,000. That's only, it really is much more than that, the number. So it's really a big number. So about 20 years ago, people is coming up with some, due to the development of mass spectrometry, coming up with say, why not if we are able to analyze and quantify, identify all these complicated lipids. Of course, in this case, we can dynamically study the lipids. How are they functioning? How are they changing? How are they interact with others? So it's leading to the emerging of the a new discipline called the lipidomics. So we are, as shown here, is the first calling this disciplinary, and it's in JOR, and it's defining what really is lipidomics. We believe it's the scope is. And of course, immediate question at the time, and still it's kind of existing. People ask, so why need lipidomics? Since lipidomics is a sub disciplinary of the metabolomics. Metabolomics has been studying the long time, and the tools is being developed, and some kind of study is a lot of advantages in the technologies is it's there. So why is needs the lipidomics to be separately developed? So I'm gonna bring up with this one from the 2 angles. One is physical chemistry, physics and chemistry, and the second is biology. Okay, from the physics and chemistry, actually the lipids you can see it's containing some kind of hydrophobic component, so can be easily recovered by the solvent extraction. So that's making the some way to easily analyze. So that's not really the big point to say it's needed establish a new disciplinary. And the second is, as I mentioned, very complicated. That's also, it's not the key point. The key point is that lipids can form aggregation. Okay, they can form the dimers, trimers, oligomers, or whatever. And it depends on the form of the lipids, also can form the micelle and the inverted micelle or bilayers. So since they are forming even in the solvent, they forming such kind of aggregation aggregates. So really, it's a very challenging to analyze them. So that's the one of the really, it's kind of the need to develop a separate technology to study the lipids. From the biology angle wise, metabolomics, particularly for the aqueous phase metabolites, usually it's studying the metabolomic study is uniform flow. It means that if you understand that we would like to study a product, and then the precursor or the substrate usually defined. And also the enzyme is one enzyme usually kind of involved. So very different from this kind of metabolites. Lipids, it's totally different. And the one node, like shown here, the sphingolipids network could involve thousands of species. And the more complicated is multiple enzymes usually involving in one pathway, as well as for like the glycerophospholipids, within the class of lipids or the category of lipids, they are remodeling. So you determine something is changed, even significance, did not tell you anything about the the flow and about the underlying mechanism or the metabolism some way. So really, it's needed to do a very in a different way to analyze, to interpret the data. So this came up, of course, different technology is being developed. Majority of the technology is separated into the two kinds. One is direct infusion based, called the shotgun lipidomics, and another type is the AOC mass based. So we developed a platform called the shotgun lipidomics. It's distinguished by the based on the multi-dimensional mass spectrometry analysis. So this type of platform comprised of this kind of component, using the multiplexed way to prepare the samples, such as the derivatization, different solvent system, etc. So it can first step to achieve the separation, and then based on the charge difference, is particularly its kind of category here, and using the ion source to separate these different lipids, and using the different variables of the mass spectrometry to analyze, identify the lipids and quantify and the data processing. So here is an example we did. The mass spec showed here, it's represent the analysis from the total mouse liver extraction. So analyzed in the negative ion mode, it show up like this kind of spectra. And if adding a little bit of mild base, such as ammonium or lithium hydroxide, something like that, and the spectra you can see it's very, very different. Particularly, I would point out is these two spark in the internal standard and the opposite, and they represent about a 50-fold separation. And then turn into the positive ion mode, and then you can see all the negative charge carried charge carried lipids gonna become the waste, cannot flow to the mass spec, and the left all the mass spec detected. And in this case, of course, you will ask, so how means this spectra or the peaks? So I'll give you a little bit examples, like the low bounded lysolipids, how it can identify and quantify the individual species. And we, of course, learned from the authentic lipid species. I need to point out lysolipids is more complicated by the isomers. It's a SN1 SO and versus SN2 SO. And then we learned that the fragmentation from mass spec analysis, it's coming up like this kind of one. It's a very abundant peak, loss 59. It's a color match the head group here. Okay. And the loss very strong abundant loss 205, the same as phosphocholine. And the unique of the two isomers is 104 versus 147. So that's the kind of the pattern. It's always there for all the lysolipids, lyso-PCs. So now we conducted some kind of the analysis. Neutral loss 59 corresponding to here, and the neutral loss 205, precursor 104, precursor 147. And you can see all these spectra so nicely selected all the lysolipid species compared to the full mass surface scanning, and so noisy, you don't know which peak is represent the lyso-PC. And now you can see the spectra. Of course, I would like to point out is that like neutral loss 59 detected peak not necessarily represent the lyso-PC, but the lyso-PC is always can be detected by such a spectra. But it combines the two of these, and the specificity increase definitely to 100%. Okay, it's both, it's very high, it's for there. And using the last two to define the isomers, and with all these spectra, and plus the full spectrum can quantify that. Then it's the question is, if the mass spec cannot pick the compound, the lipids, how to analyze them, or they are very low abundant. And then, of course, our shotgun lipidomics approach is using the different methodology to enhance the analysis with the different approach. It's like listed here. So particularly, it's very useful, very powerful is the derivatization. And so based on the functional group, and then of course can analyze its way. So we published, we developed many methodologies towards to this kind of approach through chemical derivatization. I do not have time to talking more about that. Okay, so to the current development stage, we are still kind of developing for the platform wise. We are able to the platform allow us to analyze, means identify and quantify about 30 or 50 lipid classes, 95% of the total lipid content, thousands of individual lipid molecule species, and with a minimal amount of source material with high precision. Okay, it's above 90%. Usually it's achievable to 95%. Not like in the field, people say 40% is tolerant and 20% is very good. So we can achieve basically the 5% variable or 10% variable with minimal batch effect. Here are the list of the class of lipids we are able to analyze. The green the highlighted ones with green is recently developed. So it's really kind of represent a very unique lipids being analyzed. So with this kind of development in the field, of course, move to study the different areas, including the single cell analysis and the spatial lipidomics, clinical lipidomics. And one of the front in the lipidomics study is functional lipidomics. So functional lipidomics, keeping in mind, that's different from to study the lipid function. Lipid function study, cellular lipids, what kind of the function of the class of lipids belong to the lipidology. Okay, functional lipidomics studies the lipids in the system. So it's using the lipidomics approach to identify what lipids is changed in the context of a disease, for example, or a gene knockout, something like that. And then identify the underlying mechanism leading to this kind of lipids change. And also studies what's the biological consequences of this lipid change can leading to, and also whether this kind of target identified from the mechanistic study and can serve as a drug target. So overall, that's the 3-4 buzzwords in the functional lipidomics. Okay, so my lab has been conducting this kind of study for over a decade now. It's for a long time, quite a long time. So we're using this kind of approach, studying the Alzheimer's disease, studying diabetes, study aging, study brain trauma, as well as we have lots of collaborations. The project is doing this kind of using this kind of approach. So now is the rest of the timing, I'm gonna share some results on the Alzheimer study, how we do this kind of functional lipidomics study. Okay, so Alzheimer disease is a neurodegenerative progress neurodegenerative disease, and there exist multiple hallmarks. Shown here is the A-beta peptide enriched plaques, or the tau tangles as the neurofibril tangles with the enriched with the tau and hyperphosphorylated tau, and glial activation it's very obvious and very apparent and leading to the neural information. And as well as usually you can always see if from the presentation or comparison, the AD brain have a big hole in the middle, so it's ventricular enlarged. And people don't understand why it's, but only just thinking it's just belong to the neuronal cell death. Once the cell death leading to such a big hole. So we, the clinically, and there's a lot of symptoms also can be, it's based on to classify the stage of the AD. So I would like to point out one of the symptoms is a loss of the bladder and the bowel control. Since we found some kind of connection in the our animal model study. So around about 2 decades ago, so we using the mass spec did some kind of analysis from the postmortem brain tissues of Alzheimer subjects. And obviously you can see the cluster of peak shown here, and it's disappeared in this spectrum. And of course, some kind of variable, but overall, it's changed a lot. It's lost dramatically. And the mass spec allow us to identify these ions. This compound is corresponding to the sulfatide, a class of myelin lipids. The central part is ceramide adding a sugarine, here the galactose, and the sulfate connected to that. And the enzyme, the last step synthesizes sulfatide is called CST. And the gene encoded this enzyme is GAL3-ST1. And so the first thing we thought, try to study is such a loss, remarkable, the marked loss of sulfatide. What's the biological consequence? Whether anything connected to the Alzheimer's disease. So we generated 2 line of the animal models, and the full body knockout, as well as the cell-specific knockout in the myelin, in the CNS. It means the oligodendrocytes, things like knockout. So we found it's a very interesting stuff. And using these animal models, one of the finding is the glial activation. So that's kind of a match with this hallmark. What's showed here, as you can see, is the reddish color increased dramatically in the knockout. Here it's staining for the astrocytes marker, GFAP. Okay. And also the greenish one, you can see obviously increase also very remarkably. And the represent the since that's the EBA staining represented microglial. We also did the RNA-seq type of study, it's a nanostream technology, and analyze the genes it's changed. And the showing here is how the upregulation of microglial genes specific to microglial at a different time point, as well as the different brain region. And you can see the it's a lot of upregulation is related to the disease associated as a microglial. So it's compared to the controlled one. And the animals, it's a loss of cognition, or it's not as a loss, the cognition is decline, okay? And also leading to the accumulation of the tau tangles. And you can compare to the wild type one, look at the greenish spots, and the knockout have the much higher content spots in the 7 months, and the 16 months, it's much more compared to the relatively younger age, and compared to the controls. So that's kind of the comparable with the tau tangle development. And we see the sulfatide leading to the hole without brain neuronal neural cell loss as well as the brain shrinking. But it's already sees such a ventricular enlargement. And also another piece of finding is we see the urinary bladder enlarge. So it's a loss of control of the pee and poo for these lying animals. So in summary, actually, we have more kinds of findings, such as disrupted A beta metabolism and also disrupted the cholinergic system. And the insulin resistant peripheral wise, and have some other studies, some other findings we are still going on. Okay, so then next question, of course, for the functional lipidomics is what really is the mechanism leading to this kind of sulfatase loss in Alzheimer disease. So from the study time at time, and we found maybe the risk factors for AD could be the factor, okay, could be the mechanism leading to the lipids loss. So we spend made a lot of effort to try to see whether this AD risk factors, including the genetic ones and as well as environment ones, can result in sulfatase loss. We did of that, turned out it's really is very remarkable. It's true, and show here is different A beta overproduction animals, different animal lines, and you can see the dramatic loss of the sulfatide depends on the animal model. And this kind of finding being replicated by many studies in the literature, and using different tools and to use different animal models. And ApoE4, obviously, we found is ApoE4 compared to the ApoE3 as well as mouse ApoE. It's a much lower in the ApoE4 carrier as transgenic mouse brain. And in contrast, if knock out the ApoE and the sulfatide is accumulated in the brain, so that's kind of indicate ApoE4 could have been, involving the sulfatase loss, modulating the sulfatase content. Aging, as we mentioned, that's really is a very robust risk factor. And people already did done some studies very early time as aging sulfatase is lost. And interestingly, what is summarized actually in the literature, sulfatase level in this kind of way in the human being. Around before the 30, it's due to the development of the myelin, sulfatase content in the brain is increased. And then in the middle age, it's kind of the plateau. And the beginning around 65 started to lose. Okay, so 65 is a very critical window for the Alzheimer development. So it means 65 initially started a decline of the sulfatide, combined with the other risk factors, and then could dramatically leading to the dramatic loss. We thought that was likely, the mechanism. And the diabetes, we found it's all the type 2 diabetes animal models can lead into the sulfatase as loss. Showed here is both imaging data. Okay, you can easily see the intensity, the white signal is lost, as well as the extracted lipids from electrospray analysis. The red line, the arrow indicated the loss of that, and so forth. Mechanistic wise, of course, due to the mitochondria, and also the peers also found air pollution can causing the sulfatase loss, as shown is the control versus the treated one. The yellow signal is very abundant. The very obviously is lost. So we put together, and in summary, it's of that really we believe that's we found that from our study, all the risk factors can lead to the sulfatides loss. And of course, if sulfatides loss to a certain threshold, and then it's based on our study I mentioned earlier, and leading to the AD-like symptoms. So that's likely the mechanism for why sulfatides is dramatically lost in Alzheimer's disease, and what's the connection with Alzheimer's disease. So the last point I would like to make, we believe sulfatides loss could be used as a drug target. So we have a project is try to using such a way, of course, as molecular or using a different compound to do the study. Targeted instead of the individual risk factor, we already know lots of drug companies and try the different way to study the testing the drugs and the field, particularly like the ApoE4 and the A-beta type as a targeting. And so we believe it's that's the connected one, the node would be served as a great drug target for the cure the Alzheimer's disease. All right, so I would like to acknowledge my lab members contributed to my talk, and particularly many of the former lab members, they are contributed very dramatically, very lots, done the lots of work. And a few of the collaborators related to what I presented, and the fundings from the NIH as well as the private foundation. Thank you all for your patience, for your attention. Thank you.
All right. Thank you, Dr. Han, for that insightful presentation. We'll now have about 10 minutes for questions, and I'd like to remind everyone that you may submit your questions using the Q&A feature if you haven't done so already. All right. And we'll start with, so one question, Dr. Han, is I was wondering how to prevent protein contamination in MDMSSL.
Say again, I did not catch that.
So the question is, I was wondering how to prevent protein contamination in MDMSSL.
Oh, the protein is being eliminated from the sample preparation. So it's a protein usually it's located in the middle layer of the between the aqueous phase and the solvent. So it's really it's not an issue for that. We is more worried is during the extraction sample preparation is the inorganic component.
Okay. Thank you, Dr. Han. Another question is, you listed post-operational dementia unpublished data as contributing source to sulfatide loss hypothesis. Could you please explain more on the post-operational dementia data?
Yeah, so we did some kind of the post-operational means for us, it's more like the anesthesia user. You're taking the it's a numb the animals for a long time. We see after the this kind of treatment or this kind of treatment and is leading to the sulfatide loss. So that's really not specific for operational kind of dementia, but indicate if the operational it's taking a long time with the anesthesia and it could leading to the sulfatide loss.
All right. Another question that we have is, can the sulfatide lipids be supplied in diet or not?
Yes, that's definitely, but unfortunately, the sulfatide supply, the sulfatide from dye cannot cross the BBB. So it's we have the other way to try to enhance the sulfatide in the brain.
Then another question you have is someone says, Dr. Han, this might be a stretch. I wonder whether downstream glucose-spindosine has any involvement in AD.
Glucose sphingolipids is usually involving, actually, it's a very interesting question. I would say it's more involving the Parkinson's disease than the AD. So we did not see like the sphingolipids, glycerol sphingolipids is changing. And although it's some group reported the gangliosides have involved like the Alzheimer's disease. We have not done the ganglion side kind of study.
All right. Another question you have is, how are sulfatide levels regulated in different cells? Is it similar metabolic pathways which are modulated in the different disease states?
That's it's good. Good question. The sulfatide metabolism involve 4 genes. Okay, one is the CST I showed you here, shared with you. It's the one of the synthesis involving synthesis pathway. Another synthesis pathway is a FA2H related to the fatty acid acylation. And the degradation enzyme is called ASA. And also the transport protein I showed here is the sulfatide, the ApoE. So really it's kind of the, I try to just give you some information, emphasize for the this study is these 4 genes, 3 of the AD risk gene, actually. Okay.
All right. Another question you have is, do you know the mechanism for how CST/sulfatide loss leads to enlarged bladder? This is such a fascinating connection to
We
say.
did it. Okay, I did not mention that. I showed you the observations. So each observation, we in our publication actually have the mechanistic kind of study showed what really is going on. The sulfatide loss leading to this kind of phenotype, AD-like phenotype, or why this kind of risk factor leading to the sulfatide loss. So the mechanistic kind of study is in our publication. If you are interested, you can look at these references. And so the bladder enlargement, more specifically, is all the myelin lipids. It's lost in Alzheimer patient as well as the animal. So after the sulfatide is lost, so it means sulfatide loss can leading to the other myelin lipids loss. So once the myelin lipids is lost to a certain degree, they lost the control. Of course, the spinal cord is the controlling the lots of peripheral organs as well function.
All right. Another question you have, are there any small metabolites, lipids coming out as a good biomarker for AD?
Thank you. Up to now, as I know, people, there's lots of so-called biomarker being developed and reported, and but it's kind of not commonly being used. Like our sulfatide, we can detect them from some degree as a marker in the CSF. So you know.
Right. Another question we have is, do you conduct research on the relationship between blood sulfatide levels and various disease states, particularly cardiovascular diseases?
Sulfatide in the blood is not related to the brain, unfortunately. So that's why the plasma sulfatide level is not a good marker for the brain kind of change. Since the sulfatide in the plasma is synthesized in the liver, and the brain sulfatide is synthesized, as I mentioned, in the oligodendrocytes.
All right.
So they
And then,
separated by the BBB.
all right. Then another question has, is the decrease in sulfatides related to the increase in ceramides in AD?
Ceramide, you mean the increased ceramide?
Yes.
Yeah, okay. We did found actually is from our early study at the same time, like 20 years ago, we found 3 classes of lipids actually is very remarkable, very markedly changed. One of that is the ceramide, and the second is the plasminogen, and the third is the sulfatide. So our research later on is focusing on sulfatide. The reason is that we people understand the ceramide accumulation as well as sulfatide plasminogen loss is very well, based on the mechanistic kind of connection with Alzheimer's disease. So simply say it's yes, we detected the ceramide accumulation, but not connected to the sulfatide loss. The reason is that we can easily see the molecular species profile is very different. So that's kind of one of the importance of using the lipidomics.
Okay. We have time for one more question. Actually, it's going to be a follow-up question from a question earlier. This question asks, was the CST knockout specific to the brain? Is CST also expressed in bladder cells?
The sulfatide or CST is mainly expressed in the oligodendrocytes. So other cells in the peripheral is in the kidney and the sperm, as well as the beta cell. So it's not every cell it's abundantly expressed. Of course, it's very, very minor. So you cannot see the sulfatide synthesis in these kind of cells.
Okay. Thank you, everyone, for submitting such thoughtful questions. And Dr. Han, thank you for your detailed responses. Dr. Han, I would like to hand it back to you for any final thoughts or key takeaways before we conclude today's session.
So to me, I thank you again, Taryn, and thanks ASBMB, and thank you all everyone. Joined the webinar, and to me, I would like to say is lipidomics is really is a very good tool and the technology to have to study the biology. And one of the key point I would try to emphasize is to the lipids, and we need to be more quantitative, because that way can, as I try to emphasize in my talk, could interpret the underlying mechanism and the lipid metabolism is better than the just qualitatively see the signal change. Okay, and otherwise, I also would like to mention, if anyone have any lipidomics questions, just email me so we can, discuss it, and the talking, even meetings separately. Thank you.
All right. Thank you so much, Dr. Han, for your time and expertise today, and to all of you for joining us. Be sure to keep an eye out for upcoming webinars in our Breakthrough webinar series. We hope to see you again. Have a great day.
Thank you.